Bismillahirrahmanirrahim. This is the 9700 November 2341, and we start the question 3 and 4. And in a previous video, we've done question 1 and 2. Question 3, there are more than 600 plant species in the genus whatever. Many species are grown for their attractive flowers, and some species are used as crop plants. Figure 3.1 shows the common morning glory. So you can see a picture of this, what it looks like. The gene that determines the flower color in whatever has two alleles. A dominant allele that results in purple flowers. So the dominant allele, this and this, will result in purple flowers. And a recessive allele that results in red flowers. So all the red flowers will be this. And the purple flowers can be either this, homozygous dominant, or heterozygous. A student recorded the flower cover of all the plants in a field. The student recorded 660 plants with purple flowers. So 660 with purple flowers and 440 with red flowers. So this one, the red flowers were 440. So what is the total? 660 plus 440 is 1100. Is that right? Am I wrong? The student recorded 660 with purple and 440 with red flowers. So 440 had the red flowers and the 660 had purple flowers. So we had purple and red. Assuming the Hardy-Weinberg principle applies to this population, calculate the number of plants in the field that are heterozygous, that is 2pq. Either you know Hardy-Weinberg or you don't know it. So if you don't know it, just leave it. I would rather you did a 95 mark paper and got 60 marks rather than doing the 100 marks and getting 30 marks. Use the equations p plus q is equal to 1, p square plus 2pq is equal to q square. Show your working to the nearest whole number. So number of heterozygous plants, that's a lot of work. So how are you going to do this? Let's figure this out. So q square is 440 over 1100. We just did that. So q square is equal to 0.4. So what is going to be q? Q is going to be under root. Now that is going to be rather a big figure, 0.4 under root. So we do this, and q is equal to 0.632455532. Wow, interesting. So q is this. Now we have to find out p. So how do we going to find out p? P is equal to 1 minus P is equal to 1 minus this. Why? Because this was q. So from this we take out the we find 1 minus this whole thingy, 0.632455532. Wow, that's a lot of maths. So P is equal to 0.367544468. Right? I'm sure you all have your calculators and you can figure this out. So then what are we to find out? Number of heterozygous plants. So we need to figure out what is 2pq. Now 2pq is 0.464910641. Well, what have we to find this of? Of what? Of 1100. So then the 2pq into 1100. And this will come to 511, or you could have said 512. Because it was 2pq was this. So this of 1100 into 1100. 2pq into 1100. This is equal to 511.4. So you could have said 511.4, or you can say 511.5, or you can say 511.6672. So you could have said 511, or you could have said 512. Well, I hope you can understand this. Otherwise, just leave this question. Now coming to the B part of the question, the Japanese morning glory, whatever, has over 20 different flower color phenotypes, including shades of blue, shades of purple, shades of red, and shades of pink. Wow, beautiful. God is great. So the flower color of this is controlled by at least four genes. The flower color can change gradually after the flowers open each morning and can change with fluctuations in the carbon dioxide concentration of the surrounding air. Very interesting. So they are affected by the environment. A student concluded that the flower color phenotypes of whatever shows continuous variation. Suggest two reasons why the student made this conclusion. Why? Very important. What is continuous variation? The colors are not distinct, there are no discrete categories, they overlap, they have a different range, or you can say they include many, many intermediates. So you can say a lot of things in different ways, but you have to mainly say is the color is in different categories. There are no distinct types. You can't say there are only three different types. You can't say there are only 10 different types, but they keep on changing and then they change with the environment. And environmental change is only with a continuous variation. Discontinuous cannot change with the environment. Like, for instance, I have blood group B. Now, if I don't get any food, or if I'm exposed to a lot of hot temperature, or I have been standing in the sun for a long time, my blood group is not going to change. So they don't change with the environment. So the first point was that the colors are of different categories. There are no distinct categories. There are many, many intermediates. Number two, there are more than one or there are several genes which are involved in it. Number three, environment affects the color or environment affects the phenotype. So all these things reason out that it is continuous variation. So color shows many intermediate, several genes. So this is the point where there's several gene stories here. And environment affects the phenotype. You can see this change in fluctuations in the carbon dioxide concentration of the surrounding air. Then coming to question number C part, scientists investigate the response of stomata to changing CO2 concentration in the beach morning glory, the name whatever it is. The scientists placed these plants in chambers. They measured the width of open stomata, stomatal aperture, after the plants had been exposed to different CO2 concentrations for 40 minutes. Light intensity and temperature were kept constant. The relationship between CO2 concentration and the mean width of stomatal aperture. Stomatal aperture means how wide this opening is. This is the opening. And the mean width of the stomatal aperture is shown in figure 3.2. So here we have the CO2 concentration and here we have the mean width. So at 400, the width was 2. As the CO2 concentration is increasing, is going to 1000 from 400 and is at 900. Now the stomatal aperture is only 0.5 micrometer. Here it was 2.0 micrometer. Always understand the graph like this. So at 400, what is the aperture? 2.0. At 900, it has gone down to 0.5 micrometer. This is what the unit that they have used here. So always understand the graph and then, of course, proceed with the question. Now, what does it say? It says in 2016, a study measured the atmosphere's CO2 concentration as 400 micromoles per moles. In the future, climate change may reduce water availability and increase atmospheric CO2 concentrations in some habitats. Suggest how the stomatal response shown in figure 3.2 would allow to survive the effects of climate change. Very simple. What is going to happen? As climate change increases CO2 concentration, this will cause the stomatal aperture to decrease. So stomata will open less. So less water lost, because more water will be lost through larger openings of the stomata. So this will compensate for the less water availability, because now they've told you in the question that there is less reduced water availability. So that is good. Less water available, stomata open less, so less water vapor is lost. So less water lost or less water the water that evaporates would not be lost from the leaves. So that is good for us. That's good for the plants. So let's word it now. Increase in CO2 concentration, stomata less open. That's what we've learned from the graph. This is what figure 3.2 is telling us. So less water will be lost through the stomata. And this will allow survival in low water availability, where it says here low water availability. Now coming to part 2, under certain conditions, the closure of stomata is controlled by abscisic acid. Describe how abscisic acid causes the closure of stomata. That is a direct question. Either you know this or you don't know this. Either you skipped this when you were revising or you know it. It's a very direct question. There's no ambiguity about it. There's nothing to think about it. You either know it, and basically abscisic acid binds to receptor on the guard cell surface membrane. This is the guard cell. There is a surface membrane protein receptor on its surface. The abscisic acid binds to it. So hydrogen ions stop moving out of the cell. Usually hydrogen ions are moving out of the cell. So hydrogen ions stop moving out of the cell. Calcium ions enter. Calcium ions enter the cytoplasm. This acts as a second messenger. So what is going to happen? Potassium ions will leave the cell. Water follows. Water follows. Water leaves the cell by osmosis down the water potential gradient. And so this becomes less turgid. So then what is going to happen? This is going to close. The other one is also going to close. So less turgid, flaccid guard cells close the stomata. This will close the stomata. This will come here. And there'll be no longer this thing. So the stomata close. So you have to be very careful to get your 4 out of 4 because this is a very easy part of the question. Abscisic acid binds to receptor here. H ions stop moving out. This was moving out. Now this stop moving out of the cell into the cell wall. Calcium ions enter. Potassium ions leave the cell. Calcium acts as a second messenger. Water leaves the cell by osmosis down the water potential gradient. So the cell becomes flaccid, or the guard cell becomes flaccid, or you can say becomes less turgid. And this results in the closure of the stomata. So this was happened. This is what you are explaining to me. How does a stomata close? So please understand this and give me all these points. So then we can just look at the wordings. ABA binds to receptor on guard cell surface membrane. Be very careful to write that. H positive stops moving into cell wall, or you can say hydrogen ions, protons stop moving out of the cell. Then you say calcium ions enter the cytoplasm, potassium ions leave the cell, water leaves by osmosis because now the ions that have moved out results in a higher water potential. So water leaves by osmosis, less turgid guard cells close the stomata. Then coming to the D part of the question, scientists are researching whether abscisic acid can be used in crop treatment to increase yield. Evidence suggests that abscisic acid modifies the effect of auxin on elongation growth in plants. Scientists investigated the effect of different concentrations of abscisic acid on root elongation in seedlings of Thales cress. The seedlings were divided into 4 groups. A control group, 0.0 micromole abscisic acid. 3 experimental groups, each treated with a different concentration of abscisic acid, 0.1 micromole, 1.0, and 10.0 micromole. For each group of seedlings, root length was measured for 6 days. During treatment, the rate of root elongation was calculated each day. The results are shown in figure 3.3. So we've got a whole thingy here to look at. So control 0.1, 1.0, 10.0. Now you look at the graph and you think, oh my God, we didn't study this in class. Maybe Dr. Sarah didn't teach this to us. But then most of paper 4 is something which I never taught you. So day, the rate of root elongation 200. You can see here 100, 200, 300, 400 rate of root elongation is in micrometers per hour. And we've got all these data. Interesting. Now, what do we have to look at? Now, let's look at the question. What is the question on this? We go on to the next page. With reference to figure 3.3, describe the effect of treatment with abscisic acid on the rate of root elongation. What is describe? Describe is no bio. Do not give me any biology. Describe is no biology. And these are 3 marks which every student can get because it isn't any biology. Now let's look at the graph and I'll give you the points which you are supposed to be now looking at it. First of all, what I would do is I would then put the key in here. So control is which one now? Let me look at it. So control is which one? Let me give it another color so that everybody, we are very fond of this. I love all using these colors. So which one is the control? This is the control, right? So I'll write here control. Okay, then which one is point 1? Let me give it another color. This one is point 1. This one is a point 1, 0.1. Then we have, let me use another color. Then we have the 1.0. 1.0 is the x's. Okay, so this is the 1.0. 1.0. This one is the 1.0. And then we have the triangular ones. So let me use a yellow shade for that. So the 10 micromole is this one. So this is the 10 micromole, 10.0. Now, what is the describe? Let's look at the describe. What is the answer? Point 1 gives the higher increases or gives a higher rate compared to the control. Point 1 here, you can see this one. Point 1 is more than the control. This is the control. So point 1 is more than the control, gives a higher rate compared to the control, which is the normal one. The 1 and the 10, the 1 and the 10 decrease, give a lower rate as compared to the control. So you're comparing with the control, which is the normal. Then 10 rate stays constant. The 10 rate stays constant from here to here. The 10 rate stays constant, does not increase over the 6 days. So 1, 2, 3, and this was, it really remains the constant because here you can see also it's just gone up a little, but it really stays the same. So 10 rate approximately constant plateau does not increase over time. Does not really increase a lot over the 6 days. You can see all the others are increasing. This one is increasing, this one is increasing, this one is, this nearly stays the same. And then you can do a data quote comparing the rate on 2 different days for 10 micromole. You can compare rate at 2 concentrations on 1 day. So you could do it on, I would do it on day 5 because it's a long right at the end of it. I would, give the different values for all of them. I would give the value for this one, then I'd give the value for this one here, and this one here, and this one here. And I would read off this and I would write the units so that I'd get that 1 mark for that. So please remember, a describe is just a comparison of the graph. You don't have to give me any bio. Why this is happening, how this is happening, no bio at all. Please remember this. So data quote means that you would give me figures for all these on any one specific day. You could have done it on day 5, you could have done it on day 6. So I have even come out with these. I might be wrong here, I might have done a few, I've not been very accurate in this. But the main thing is that you had to give me the units, and this is how you do a data quote, which is a very simple way of doing it. And everybody can do that. And I think you must do it with every described question. Every described question, you must give me a data quote. That's one way of doing a very sensible exam skill. Then coming to the next part of the question, part 2, the passage outlines the role of oxygen elongation growth in plants. Complete the passage by using the most appropriate scientific terms. The binding of oxygen to receptor causes to be pumped. What pumped out? Protons. So protons, or you could have said H positive ions, right? Proton be pumped into cell walls. This activates proteins called expansins, which disrupt the links between cellulose microfibrils. The cell walls are then able to expand. So there were 2 marks for this. One is for the protons and the other was for the cellulose microfibrils. That finishes a 16-mark question. Question number 4. The potato plant Solanum tuberosum is an important food crop. Crop yield is reduced if the leaves of the plant are eaten by the larvae, immature stages of the Colorado beetle. Crop scientists used recombinant DNA technology to create 2 genetically modified GM varieties of potato plant. These plants produce proteins that are poisonous to insects. So we put in a gene and that is going to result in producing something toxic, and when the poor insect comes to eat the leaves of the plant, the larvae or the caterpillars and poor caterpillars will die. GM variety A contains 2 new genes, SN and BT. GM variety B contains 2 new genes, SN and something OC2. The new varieties were tested by having a constant number of Colorado beetle larvae introduced to the plants at time 0 hours. The number of larvae that were alive after 24, then 48 and 72 hours was recorded. So what did we do? At 0 hour, we put a constant number of the larvae on it. And then the number of larvae that were alive after 24, 48, and 72 hours was recorded. I think that's rather being very, very nasty and mean to these insects. I think animals have rights as well. The percentage of the larva that had died in each time interval was calculated. This was repeated for potato plants that had not been genetically modified, the non-GM ones. Table 4.1 shows the percentage of Colorado beetle larvae that had died on the GM potato plant varieties and on the non-GM. So now there's a lot of analysis of data. So we have the type of potato plant A, B, and the non-GM, and then in 24 hours, percentage of color that had died. So 50% had died, 37% had died, and 0% had died. 48 hours, 93 had died, 70 had died, and 0 had died. Then at 72 hours, 100% had died, 93% had died, and 0% had died. Non-GM ones. Suggest what is meant by recombinant DNA technology. That's 2 marks only. So what is recombinant DNA technology? Basically, what is it? It is DNA from 2 different species. And using enzymes or vectors, we create a transgenic organism or a genetically modified organism, like we take the insulin gene and put it into bacteria. So what is recombinant DNA? Suggest what is meant by recombinant DNA technology. So the DNA is joined, made from 2 different species, like in the human insulin gene is put into bacteria, so it's 2 different species. And we use enzymes like we use DNA polymerase, restriction enzymes, or we use vectors to create, put it into plasmids. So we use vectors to create a transgenic organism. So suggest why the scientists created 2 different types of GM potato plant so that we could compare the effectiveness of the different genes, the BT and the OC2 plant varieties A and B, to look to which gene variety A or B works better, which one is more effective in killing the insects. So because if then we don't need to use any insecticides and these insects would die. Then it says state why the scientists also perform this test on the non-GM potato plant to compare or to work as to use as a baseline or to be called as a control. That is what we do in a control. We have something to compare it with. Like if I say pizza with olives is tastier. So okay, then we have 2 pizzas, one with olives, one without olives, and we see which one is tastier. So we have to use it as a control. So DNA joined from 2 different species using enzymes, or you could have said using vectors to create a GMO, which is a genetically modified organism, or you could have used the word transgenic organism to compare effectiveness of genes. This one is this part, and then the third is state to compare or to work as a control. Then coming to the B part of the question, which has 3 marks, always look at the marks first and then decide what you're going to write. Discuss how the results in table 4.1 provide information that could help to solve the global demand for food. Well, A and B kill larvae. So if they kill the larvae, then this genetically modified, they will increase the yield of food from potatoes. So this genetic modification will increase the yield of food from potatoes. So A is the best, kills most of them. So should be grown and will provide a lot of potato. So will provide a lot of food for the human beings living on the face of this earth. So A and B both result in killing the larvae, which is going to destroy the leaves. So the leaves, less leaves means less photosynthetic area, means less glucose, means less starch, means less crop yield. Now these genetic modification is increasing the yield of food from potatoes. And because A was the best, because it killed all the larvae, so will provide the most crop yield, most food, or you can say more potatoes, so more food for the human beings which live on the face of this earth. Question 5, the respiratory quotient RQ values to different respiratory substrates can be calculated. Table 5.1 shows the formula for 4 respiratory substrates, the number of oxygen molecules needed to complete respiratory substrate to carbon dioxide and water. Using table 5.1, name the respiratory substrate with the highest RQ and the respiratory substrate with the lowest. So highest RQ was malic acid. This is something very basic. Malic acid has the highest RQ. Why? You have to do it. How will you do it? C6, you have to do it CO2 over O2. And the lowest was oleic acid. So as we all know, it's CO2 over O2. So C was the here, but it's written C4, C6, C4, C18. So you just have to divide that by O2. O2 was given to you here. It was given in this table. And that is why it's malic acid and oleic acid. Then we come into the next part, which is in anaerobic respiration. In anaerobic condition, the production of ATP in mammals and yeast involves glycolysis, not fermentation. Describe the similarities and differences between fermentation in mammals and in yeast. Now that was very simple. There's a 5 marks for that. Very direct question. Similarities: use pyruvate, occurs in the cytoplasm, makes NAD, or you can say regenerates NAD, and it's a redox reaction. Differences: lactic acid made in mammals, ethanol in yeast. One step in mammals, two steps in yeast. Carbon dioxide made in yeast, not in mammals. Reversible in mammals and irreversible in yeast. So, quick recap, similarities: use pyruvates, occur in cytoplasm, regenerate NAD, redox reaction. Differences: I made a column, mammals in yeast. You can always do that. Nowhere does it say you cannot make a column. So, lactic acid in mammals, ethanol in yeast, 1 mark, one step, two step, second mark. CO2 made, not made, third mark, and then reversible and not reversible. So that completes this here. I'm going to continue the rest of these questions in the next video. Thank you very much.